Hello Year 13, Mrs Drage again here. Right, going to explain to you now about how an anaerobic respiration works. You'll be very glad to hear that it's relatively straightforward given the work we've done so far. And I will just talk to you a little bit on the end about how we can use respirometers to measure the rate of respiration. Specifically that, knowledge of that is not required, but it's a classic application question, so it always helps to go over those things in advance. Okay, so we'll have a quick chat first of all about anaerobic respiration, the different forms that it can take. So we're talking about the eukaryotes. There's plants and some of the microorganisms, obviously yeast being the prime example of that, are actually able to take pyruvate, which as you know is the end product of glycolysis, and convert it into ethanol and carbon dioxide. Okay, so yeast is pretty cool actually because it can do both aerobic and anaerobic respiration, depending on the availability of oxygen. So we call those things partial anaerobes. Okay, and the other type of anaerobic respiration that we come across is in animals where they convert pyruvate into lactic acid, which is something you would be probably more familiar with from your work at GCSE. Okay, and obviously it's lactic acid that goes on to cause us fatigue in our muscles, which I'll chat a little bit more about in a while. Okay, so getting down to the detail of this then. So obviously we're aware that anaerobic means without oxygen. So if there's no oxygen, then there's no terminal electron acceptor. So the electron transport chain cannot function. The hydrogens that have been given to the carriers, or would have been given to the carriers, remain on the carriers. They remain reduced. No further respiration can happen. No further aerobic respiration can happen. This means oxidative phosphorylation has stopped. So we have to go back now and look at glycolysis, where I'll explain to you how animals, organisms are actually able to get the energy that they need from the first stage. Okay, so as you can see here, you'll recognise the stages of glycolysis. So we've got some glucose, activated by a little bit of ATP, and converted into pyruvate. So that's happening in glycolysis. And you should remember that we also at that stage generate some of our reduced coenzyme NAD. Okay, so that, if you remember, is actually made from where triose phosphate is converted into pyruvate or pyruvic acid. Now, the cool thing is that obviously we, first of all, generate a little bit of energy. Not very much, admittedly, but it's enough just to get us by for a little while. But also, we've got this reduced carrier, this NADH. And actually, what we now need to do if we want this process to continue in the cytoplasm is to get our carrier and convert it back into NAD. Okay, and the way that we can do that, well, the two ways that we can do that, what I'm showing you here is anaerobic respiration in animals. So we can see that we're going to make some lactic acid as our end product. Okay, so what actually happens is when you convert pyruvate into lactic acid, you actually oxidize the NAD. So the NADH, sorry. So you oxidize it and convert it back into NAD. So this process here is a reduction. Pyruvate is reduced into lactic acid. And in that process, NADH is oxidized. So obviously, again, it's a redox reaction. And that enables this whole process to continue. So this oxidized NAD is then able to feed back in to the reaction where glucose is broken down into pyruvate. And so we continually get this cycle. Okay, so just to show you this in a slightly different format now, everything's exactly the same as I just explained on the previous slide, just a different way of looking at it. So over here, you can see we've got our glucose. So this is glycolysis step. And as we know from our work on aerobic respiration, the first stage converts glucose into the 3-carbon pyruvate. In that process, we know we generate a little bit of ATP, which is obviously what's being harnessed and used in aerobic respiration. That's the whole point. And that would make a little bit of our reduced carrier. And as I mentioned on the last slide, we need to regenerate that because otherwise this whole process will stop. Okay, so the whole point of the next step, converting pyruvate into lactic acid here, is that we oxidize this carrier, this coenzyme, back to NAD. which can feed back in here. And so we keep looping around, okay? So without that regeneration, glycolysis would also stop. So now we really are in trouble, okay? So there'll be absolutely no energy whatsoever, and of course that's the end. Now this is a very short-term solution, okay? You'll know yourselves that you can't do anaerobic activity for very long. The lactic acid that builds up is not particularly great for us. It causes quite a lot of muscle fatigue. Reasons why, again, outside of the spec, but obviously it's lactic acid, it's acidic. We know what acids do to proteins. They don't particularly like being in them. They'll change their shape. Muscles are made from proteins. Again, it's outside of the spec, but ultimately it affects how the muscle fibres slide over each other and causes them to fatigue. And you end up with, from GCSE, I'm sure you can remember, the oxygen debt, which means basically you have to pay it back. You have to, when you finish doing anaerobic activity, you need to break this lactate down. Okay, and basically what happens is it gets converted back into pyruvate, and then it can go off into the link reaction and Krebs, so that we can then continue breaking down the original glucose properly by oxidative phosphorylation. Okay, it's actually possible, if it's not sent off to Krebs, that it could actually go off and be stored. So it can, there's actually a biochemical pathway where it can be stored as glycogen. But it's probably easiest to imagine that once we convert that lactate back, it will go off and be involved in the Krebs cycle via the link reaction. Obviously, when we convert it back to pyruvate, that's an oxidation step. So our coenzyme has been reduced, and again that's free to go off and be used in the ETC. Actually, I've pointed that up there, but I mean to the electron transport chain. So we've got our reduced NAD back that we originally used up, that came from glycolysis. So you can see here on this one, obviously we've got the alcoholic fermentation up at the top, lactic acid process that we've just talked about, but we've also got on this slide alcoholic fermentation, which is what I mentioned at the beginning, that yeast can do. So in yeast, as an example, instead of converting to lactate, they convert to ethanol, which is a 2-carbon substance, and in the process release some carbon dioxide. But the essential, the principle, the point of this chemical equation is the same in both cases. And the whole idea is that you are regenerating your NAD so that you can basically keep this process going. So just so you can sort of see how that all kind of fits together, obviously at the top here, got our straightforward aerobic respiration that we've been talking about in the other unit, and obviously you can see there that theoretically, I know you're all laughing at me saying that, but theoretically you can make 38 ATP molecules from fully breaking down glucose in the presence of oxygen. But actually, when you start, when you remove the oxygen and you make it go anaerobic, as you can see, hugely inefficient. So breaking down a whole glucose molecule is only going to get you 2 ATPs. And as we know, they come from that stage in glycolysis. Okay, but remember, we can't continue with that unless we regenerate the NAD by breaking down the pyruvate into either ethanol, in the case of plants or some microorganisms such as yeast, which is the example I gave, or lactic acid in animals.